Hi everyone, in this recording we'll learn a Bacteriophage mu. The Bacteriophage mu is a temperate phage like lambda phage. That means it has a choice to undergo either a lytic cycle or a lysogenic cycle, but has the unusual property of replicating by transposition. What are transposable elements or transposons? They are sequences of DNA that can move from one location on their host genome to another as discrete genetic units. They can jump from one place to another on the DNA. That's why they are also called as jumping genes. Why are we talking about transposable elements and jumping genes when we are learning a bacteriophage? The reason is mu is both a bacteriophage and a very large transposable element that replicate its DNA by transposition. So we have learned so many bacteriophages. What's so special about this one? That this bacteriophage is a transposable element that replicates its DNA by transposition. Transposable elements, when they jump from one place to another, can generate mutations. And mu also does that. This is the reason it's called as mu. The phage was named mu because it generates mutations when it integrates into the host cell chromosome. The mutations result because the mu genome can be inserted within the host genes, interrupting the genes or coding sequences. Hence, a host cell that is infected with mu will often gain a mutant phenotype, and mu is a very useful phage in bacterial genetics because it can be used to generate bacterial mutants. It's used as a biological mutagen. Let's see some basic properties of bacteriophage mu. Bacteriophage mu is a very large virus with an icosahedral head, a helical tail, and 6 tail fibers. The genome of mu consists of linear double-stranded DNA. So it's a DNA virus having double-stranded DNA as their genetic material, linear. Most mu genes are involved in the synthesis of head and tail proteins, and important genes at each end of the genome are involved in replication and host range. This is a genetic map of bacteriophage mu. The mu virion contains approximately 39 kbp or kilobase pairs of DNA. But actually, only 37.2 kbp constitute the actual mu genome. So what about the rest? The additional length is the host DNA attached to the ends of the mu genome, 150 base pair at the left end and 1 to 2 kilobase pair at the right end is the host DNA. So the mu genome is 37.2 kbp, and flanking on both the sides, left end and right end, there is host DNA, making it the total length 39 kbp. These host DNA sequences are not unique, but merely represent DNA adjacent to the location where mu was inserted into the host genome in the previous host. How does this happen? When a mu virion is formed, a length of the DNA containing the mu genome, just large enough to fill the phage head, is excised from the host. The DNA is packaged until the head is full, but the place at the right end where the DNA is cut varies from one virion to another. For that reason, there's a variable sequence, host sequence at the right end of the phage. This is host DNA right of the at R site, which is this one. And each virion arising from a single infected cell is genetically unique in having different flanking host DNA. When a bacteriophage infects a host, after the cycle is complete, many bacteriophages comes out of that host after lysis. Here, each mu virion is genetically unique. It's different, having a different host sequence at the right end. That's why it's called as variable end. After the properties, let's talk about an invertible region of mu. As shown in this genetic map, a specific segment of the mu genome called G is invertible. Now, this is the G region which I have circled in the genetic map or the genome. This region is invertible. That's why called as invertible G region. It can present in the genome, either in G plus orientation or G minus. So there are two possibilities. This G region will be either in the G plus orientation or G minus. The orientation of this segment determines the kind of the tail fibers that are present on the phage. So if I draw the bacteriophage here on this slide, this is the icosahedral head, this is the helical tail, and these are the tail fibers. Now these tail fibers are important in attachment of the phage to the host cell. And this G segment is responsible for which tail fibers will be present on the phage. So if does the phage have the option of having a different tail fiber? Yes. Because adsorption to the host cell is controlled by the molecular interactions between the tail fibers and the receptor site on the cell surface, the host range of the mu is determined by the orientation of the G segment. So the G segment is in the G plus orientation or G minus will determine the kind of the tail fibers that the phage has, and that determines the host range. Okay, whether the G segment is in G plus or G minus will determine the host range. If the G segment is in the G plus orientation, the phage will make tail fibers that allow it to infect E. coli K12. If it's in G plus, the phage will make tail fibers and can infect E. coli strains K12. What if it is in G minus? In contrast, if the G segment is in the G minus orientation, then the phage will make tail fibers so that it can infect E. coli strain C and several other species of enteric bacteria. So whether this bacteriophage mu can infect E. coli K12 or E. coli C and other species depends on the tail fibers, and that depends on whether G segment is in G plus orientation or G minus. So this invertible G can be present in either G plus or G minus, and that determines the host range. The two alternative tail fiber proteins are encoded on opposite strands within the small G segment. Left of the G segment is a promoter that directs the transcription of the G. In the orientation G plus, the promoter for the genes S and U is active. So if the G segment is in the orientation G plus, a promoter is active for genes S and U and they will be transcribed. Whereas in the orientation G minus, a different promoter directs the transcription of S prime and U prime on the opposite strand. Inversion of the G region is a rare event and it's under the control of another gene that determines or decides the inversion, and this gene is adjacent to the G region. This inversion phenomenon is a simple mechanism or strategy that the phage has evolved for attacking a variety of different host cells. Let's see the replication of mu. T4, T7, lambda phage, mu, all are DNA bacteriophages, DNA viruses, all have linear genomes. It's very difficult for any organism to replicate a linear genome. Eukaryotes have telomerase for replicating the ends of the genome. These viruses use distinct schemes to replicate the end. T4 and T7 use terminal repeats to form concatamers, whereas lambda circularizes its genome after infection. Mu replicates in a completely different manner from all of these because its genome is replicated as part of a larger molecule. It's integrated. On infection by a host cell by mu, the DNA is injected and it's protected by host restriction or restriction endonucleases by a modification system in which the adenine residues of mu are modified by acetylation. In contrast with lambda, where the integration is required only for lysogeny, integration of mu DNA into the host genome is essential for both lytic and lysogeny. Whether mu enters via lytic or lysogenic, integration is essential. Integration requires the activity of a gene A product, which is a transposase. A 5 base pair duplication of the host DNA or target site is generated where mu becomes integrated. So this is the host genome. This is the target site region to be duplicated. And this duplication arises because staggered cuts are made, which you can see here in this picture. In the host genome where mu will be inserted, staggered cuts made by the transposase in the host DNA region to be duplicated. The resulting, and after the staggered cuts are made, you can see how mu is inserted, and this target site or where the mu got inserted, one strand is here, another is here, and when these ends are or gaps are filled, conversion of single strands and insertion into mu, insertion of mu, and when these gaps are filled, repair of this DNA leads to the formation of a 5 base pair duplication of the target site. Duplication of short stretches of host DNA is typical of transposable element insertion. Mu can enter the lytic pathway either upon initial infection if the repressor is not made, or by induction of a lysogen. Now we all know for lysogeny a repressor is needed. This lowercase c gene codes for the repressor, and mu can enter the lytic pathway either upon initial infection or if this repressor, which is product of the lowercase c gene, is not made, or by induction of a lysogen. In either case, mu is replicated by repeated transposition of mu to multiple sites on the host genome. And initially, only the early genes of mu are transcribed, but after the capital C or the uppercase C protein is expressed, which is the positive activator of late mRNA synthesis. So initially, only the early genes are transcribed, but after this uppercase C or capital C protein is expressed, which is a positive activator for late transcription, the mu head and tail proteins are synthesized. Eventually, the cell is lysed, mature phage particles are released, and this is lytic cycle. For the lysogenic state in mu, it requires a sufficient accumulation of the repressor protein, which is the product of this lowercase c gene. So for the lysogeny, mu requires a sufficient accumulation of repressor protein to prevent transcription of the integrated mu DNA. In this recording or video, we have studied mu bacteriophage, introduction, basic properties, invertible G region and its significance, and the replication.